Hello friends, welcome to BMH Learning. In this video, we will be discussing about S1 nuclease. First, let's remind, what is a nucleus? A nucleus is an enzyme capable of cleaving the phosphodiester bonds between nucleotides of nucleic acids. Simply put, a nucleus is used to cut the DNA. Now, this cut can be done both on single-stranded as well as double-stranded DNA. Now, talking about the S1 nuclease, this enzyme is a nuclease that hydrolyzes nucleic acids into 5' mononucleotides. Now, the important feature of this enzyme is that it acts on both forms of nucleic acids, that is DNA and RNA. But the specificity of this enzyme is such that it only acts on the single-stranded version of both DNA and RNA. That means it splits only single-stranded DNA and RNA. Although its primary substrate is single-stranded, but there is something more. Occasionally, it can also introduce single-stranded breaks in double-stranded DNA or RNA, or you can say DNA-RNA hybrids. Though S1 nuclease can act on double-stranded DNA or DNA-RNA hybrids, but it would still hydrolyze single-stranded region in the duplex DNA, which are present in the form of loops or gaps. Let me tell you one more thing. This enzyme is an endonuclease. That means it cleaves the phosphodiester bond within a polynucleotide chain. In simple words, it makes a cut within the DNA length. Now let's talk about mechanism of S1 nuclease. So we are given a molecule of double-stranded DNA, which has already been digested with EcoR1. The treatment with EcoR1 resulted in the formation of the sticky end. Do you know, the sticky end is a single-stranded region of DNA. Now let's treat this DNA molecule having overhanging end with SN1 nuclease. So the enzyme specifically will choose single-stranded overhanging end and will cleave phosphate groups from 3' nucleotides right here. And what we are going to get? This. And these are 5' nucleotides. Now let's talk about some properties of this enzyme. S1 is a monomeric protein of a molecular weight of 38 kilodalton. To function properly, this enzyme requires zinc ions as a cofactor. The enzyme has a very important property. It is relatively stable against denaturing agents like urea, SDS, or formaldehyde. This is the optimum pH range for S1 nuclease activity. The enzyme can be inhibited by a certain concentration of particular molecules. Like 1 millimolar ATP can completely inhibit the enzyme. Also, 50% inhibition has been shown at 85 micromolar DAMP. Now, there are very important applications of this enzyme. The enzyme is used in nuclease mapping techniques. It's very useful in the removal of single-stranded regions from double-stranded DNA, as we saw in the mechanism. SN1 is also used in opening hairpin loops generated during synthesis of double-stranded cDNA. It's a very powerful tool as a reagent in nucleus protection assays. This was all. Thanks for watching.